or viruses, probably until now.
Okay, we will start transmitting in YouTube in a few seconds.
Okay.
Listo, me parece que ya estamos en vivo. Entonces,
Okay, we will start transmitting in YouTube.
Entonces, les damos la bienvenida a nuestra conferencia de hoy de astrobiología para todas las personas. El día de hoy tenemos a un excelente invitado y la doctora Irma Lozada nos va a ayudar a presentarlo. Por favor, Irma, adelante.
Sí, muy bien. El doctor Kenneth Stedman es un virólogo de ambientes extremos, tales como los fondos marinos oceánicos y los manantiales volcánicos termales y acídicos. Él obtuvo la ingeniería química por la Universidad de Stanford y el doctorado en biología celular y molecular por la Universidad de California. Hizo estancias postdoctorales en Suiza, Alemania, Holanda, Estados Unidos, y actualmente es profesor de biología en la Universidad del Estado de Portland, en Estados Unidos. Las investigaciones científicas del profesor Stedman en virología son diversas, las cuales se reportan en alrededor de 50 artículos científicos y 20 capítulos de libros. Nos gustaría, aunque tiene muchos reconocimientos, nos gustaría resaltar 5 de sus contribuciones más relevantes. Es el primero en descubrir en el año 2012 que existe la recombinación genética entre virus no relacionados, esto es, aquellos que tienen genomas de ARN y de ADN. Ha dedicado más de 20 años de estudio a la caracterización de un nuevo virus arqueano extremófilo llamado Saccharolobus o previamente Sulfolobus, cuya estructura externa posee un diseño particular que le permite sobrevivir un ambiente natural que le es altamente acídico, en pH 2 aproximadamente, y con temperaturas mayores a los 80 grados centígrados. También demostró que la mineralización con silicatos de la estructura viral permite la inactivación reversible de estos virus extremos, así como una mayor tolerancia a la desecación. Él también creó la compañía Stone Stable para identificar moléculas y nanopartículas que ayuden a estabilizar las estructuras de ciertos compuestos, como aquellos utilizados en las vacunas, así como también es uno de los miembros fundadores del grupo de investigación de la NASA enfocado a la virología, cuyos objetivos incluyen la identificación y validación de posibles bioseñales para virus y, en consecuencia, la identificación indirecta de vida extraterrestre fuera de la Tierra. Además de todo esto, el doctor Kenneth Stedman ha asumido una responsabilidad pública de la ciencia, por lo que ha mantenido una comunicación constante con diversos medios de comunicación para entender las diversas fases de la actual pandemia del COVID-19. Como una cortesía hacia nuestro invitado y la audiencia que no habla español y que verá este video, esta plática después, permítanme hacer la semblanza del profesor Stedman ahora en inglés. Dr. Kenneth Stedman is a virologist of extreme environments, such as those found at the bottom of the oceans and in volcanic acid hot springs. He got a bachelor's degree in chemical engineering by the Stanford University and a PhD in molecular and cell biology by the University of California. His postdoc career has been developed across Germany, Switzerland, Netherlands, and United States, where he is now a full professor in biology at Portland State University. His scientific research is hugely diverse, with around 50 scientific publications and over 20 book chapters in the field of virology. He has received several awards, but we would like just to highlight 5 of his major scientific contributions. He is the first one to discover right combination between RNA and DNA viruses. He has also studied for over 20 years a new extreme archaean virus called Saccharolobus, or previously Sulfolobus, whose external structure has to survive extreme conditions of high temperatures above 80 degrees Celsius and acidic conditions below pH 4. He showed that the mineralization with silicates of the viral structure is a key mechanism to activate and inactivate these extreme viruses, as well as to tolerate desiccation. He is also the founder of the Stone Stable company, created to identify molecules and nanoparticles that could be useful to stabilize the structures of compounds for vaccines and medical drugs. He's also the founder member of the Virus Focus Research Group at NASA, the so-called astrobiology, whose main goals are the identification and validation of possible biosignatures for viruses, and in consequence, the indirect identification of alien life outside Earth. It's very important also to note that Professor Stedman has assumed an important social role in science communication by discussing and clarifying in public media the different phases of the current COVID-19 pandemic. So the Mexican Society of Astrobiology is very glad and very honored to welcome Dr. Stedman. Please, Professor, the screen is all yours now.
So thank you very much, both to the Mexican Society for Astrobiology and also Irma and Antigona. And actually, I don't need to give the rest of my talk because I think you've given it already. So at least in terms of the background. And I will try and get back to some of those themes as I continue over the next probably 45 minutes to an hour or so. One thing that I did want to mention really right at the beginning is, yes, I'm quite active, particularly on Twitter. So Extreme Virus Prof, you can see down at the bottom of the slide. And also the website from our research group, which I didn't make, it was one of my students, which means it's nice, unlike one I would do, extremeviruses.org. And there I do have a blog where I'm talking a little bit about SARS-CoV-2 and COVID and what the situation is, and mostly having to do with issues of misinformation and things that are not being properly understood. But I don't really think that it's appropriate to give any kind of talk on viruses right now without just spending the next, let's say, 5 or 10 seconds just remembering the people we have lost because of this viral pandemic. So just a couple of minutes, seconds to think about all of these things. So thank you. What I'm not going to talk about is this, killer alien viruses. So my colleagues and I, a couple of years ago in 2018, we wrote an article on astrovirology, and it got picked up by the media, and this is what they thought was happening. You know, killer alien space viruses that could wipe out humanity, maybe terrifyingly common. No, that's not what we said. They clearly did not read our paper. I will give you references later on and you can take a look at it. What I'm going to tell you about again for the next 40 minutes or so, basically is how viruses are actually really not that bad. And my feeling is viruses actually have a really bad reputation. I think we should appreciate viruses much, much more. So I'll tell you a little bit about why they have a bad reputation, and makes sense. And then I'll move on to sort of what viruses are and how I think about them. And then just some things that, at least to me, were so surprising about viruses that I just had no idea. And they're really sort of, you know, mind-blowing, the emojis that you see as, you know, what's going on with viruses. And we'll finish up a little bit talking about some of the work that we've done, just very briefly, and also a little bit about some of the work we're thinking about in terms of viruses in space. So first, viruses are bad. The reason that viruses are bad, that's how they were defined in the very first place. They were things that cause disease. Thousands of years ago, the Egyptians saw that there was polio-like disease. A little over 100 years ago, when people were looking at plant viruses, they found these really small things, too small to be bacteria, but were still causing disease. And then they called them contagium vividum fluvium. So basically a contagious living liquid. And that's because they could go through these filters. This originally then got called virus, and virus is slime, poison, venom. So it's just pretty bad, I think. And of course, as all of us know and are unfortunately still dealing with, COVID-19 caused by SARS-CoV-2. But again, this is the original definition. And one of the other things that I like to do, one of the things that I do is I also teach virology at Portland State University, is think about what really is a virus. Yes, it's this thing that you can put through filters and can cause disease, but how do you think about it in a bigger picture? So some of the standard definitions of viruses are here. One of the ones that I like the best, although I don't completely agree with, is a piece of bad news wrapped up in a protein or wrapped up in a protein coat. Your standard textbook definition is a very small, infectious, obligate intracellular parasite. So viruses have to get their genome inside of cells in order to be able to make more virus. My standard definition I use in my classes is it's a bag of nucleic acid. Nothing about bad, nothing about infections, etc. But this is actually not really complete. And this is something I only realized relatively recently. And that is that the virus is not just the particle. And so to get this analogy, I really like this painting, which probably many of you know from René Magritte. Ceci n'est pas un pipe. Now, this is not a pipe, because it's a picture of a pipe, etc. But Patrick Forterre, one of my colleagues in France, said, Ceci n'est pas un virus. Well, what does he mean about that? Well, what he says is that the particle, what you see on the outside of the cell, is only part of really a virus life cycle. And so I much prefer the definition that Salvador Luria has, which is, and I'm sorry I'm going to read this, but I think it's really very important, is that viruses are entities whose genomes are elements of nucleic acid that replicate inside living cells using the cellular machinery, causing the synthesis of specialized elements, that's that external particle, that can transfer the viral genome to other cells. And so that's basically shown here in a classic picture. This is just from a textbook. Here we have an uninfected cell. It gets infected by one of these specialized elements. The genome of this specialized element, the virus, gets inside the cell. It reprograms that cell in order to make more of these particles. And this whole cycle goes around and around. And so when I think about viruses, I don't think about that extracellular particle. I think about it this whole process. And I think that's very important when you think about what happens with viruses. Is viruses only are going to be functional when they're together with their host. And another analogy that I like is that that particle is really kind of like a seed relative to a tree or to some other kind of plant. And so it only is going to be making the whole plant or the whole virus if it's in that right particular environment. So again, just as a big picture kind of thing. Now, the interesting thing about those particles is these particles have just amazingly unique shapes. And these are just a couple of examples of some of those shapes. The extracellular particle of a virus is called a virion, and they've got a whole variety of different shapes. Some, like dengue, have pretty much like a sphere. Myxoma, which is the pox virus-like structure, is more like a brick structure. The vast majority of viruses, and we'll see many more of these later on, have these, I like to think of them as almost like spaceship-like structures. They have a icosahedral kind of shell at the top and then a tail. These are the most common viruses that you'll find in environments, but also some just very strange ones. And I'll talk a little bit more about these later on. The SSV virions, sulfolobus spindle-shaped virus. These you find in volcanic hot springs. And probably the most amazing of these structures, one of my colleagues, David Prangashvili, found these. They're little bottle-shaped viruses. One thing also to mention here, all these bars are 100 nanometers. So they're really, really, really tiny structures. but they have these amazing shapes. And one of the things that got me really interested in viruses is how do these shapes fit together? And what is it about the shape? And particularly in some of these extreme environments, what is it about that shape which allows this virus to be able to have all of the functions and the resistances that they need to have? So first, these structures, the extracellular specialized particles, the virions, are pretty unique, but also the genomes. That nucleic acid which is being transferred is also very unique in viruses, because viruses use pretty much any kind of nucleic acid you can imagine for their genome. Our genome and the genome of all cells has double-stranded DNA. So these black lines represent DNA here. But viruses, including our probably least favorite virus right now, SARS-CoV-2, use RNA as their genomes. They can also use 2 strands of DNA like we have, or they can have just a single strand of DNA or a single strand of RNA. So they have this huge amount of diversity from the structural point of view of the actual virus particles, but also huge amounts of diversity from the nucleic acid and genome level. And as probably most of you know, many of the theories about how life arose on Earth said it was using RNA as its genome. Well, there are things on Earth right now that are using RNA as their genome, and those are the viruses. So unique shapes, unique genomes. Where are these viruses? Well, my short message here is viruses are actually absolutely everywhere on Earth and in space. And I'll give you an example of some viruses in space right at the end there. But first, while we're on this space idea, hopefully all of you know this picture. Hubble, deep field, all of these galaxies surrounding our galaxies, almost innumerable numbers of stars. We're definitely not alone as far as stars are concerned in our universe. Now, what I find to be particularly even more interesting is if you basically take the telescope and turn it around. Now, if you have a microscope and you take some seawater and just take that seawater and stain it with a fluorescent dye, which will bind to any kind of nucleic acid, RNA, DNA, single-stranded, double-stranded, and just look at it under the microscope, what you see is really fascinating. You'll see a couple of diatoms. These are eukaryotic cells, a few bacteria and archaea, the microbes in here, these larger dots. But you'll notice there are a vast, overwhelming number of these little tiny dots. And these little tiny dots, these are virions. They're too small to be cells. And if you look at them in the under-electron microscope, you see they have these shapes that really look very specifically like viruses. So you can count the numbers of these spots, and you can look at the number of milliliters of water in the ocean, and you come to some really ridiculous numbers. Pretty much anywhere you look on Earth in the oceans, between 1 million to 10 million of these virions per milliliter. And there's a lot of milliliters in the Earth's oceans. So that gets you to a really astronomical number. And people in astrobiology understand these kinds of numbers. Most biologists, 10 to the 31, they have no idea what that is. But yeah, they're really humongous numbers of virus particles. And these are infecting cells in the ocean all the time, 10 to the 13 infections per second. It's really just an amazing number of virus infections that are going on all around us all the time. It's best understood in oceans, but also it turns out these are in soils. They're really in pretty much any environment that we've looked at. You see massive numbers of viruses. And it turns out that some of them also carry some very interesting genes, and I'll get back to that a little bit later on. But first, just this, absolutely astronomical number. And this is something, it's a slide which the late Roger Hendricks gave me, using a wonderful quote, which was from J.B.S. Haldane, a very well-known environmental biologist, who said, the creator must have had an inordinate fondness for beetles because there's so many different species of beetles and there's so many of them. Well, if there were 10 to the 31 beetles on Earth, it would be this size relative to the Earth with 10 to the 31 virus particles on it. So basically, the creator must have had an inordinate fondness for viruses, not really for beetles. And also from courtesy of Roger Hendricks, is if you were to take all of these particles and line them up end to end, which of course they don't do, but if you were to line them up end to end, 10 to the 31 of these particles would be 20 million light years from one end to the other. That's 4 times as far away as the Andromeda Nebula. And this is just absolutely crazy numbers of viruses. So this is all theoretical. They're not all going to line up like that. But it turns out you can actually see virus infections from space. This is an example of a picture, again, from space. This is Cornwall, where they just had the G7 summit, is right here at the end of Cornwall. And the light blue that you see there, this is an algal bloom from Emiliana huxleyi, which is a coccolithophore, really wonderful eukaryotic alga, but has all these calcite plates around it. And so what happens is when these algae are just floating around by themselves, they're colorless. But when they get very large numbers, what happens is very large numbers then get infected by the viruses which are present in the seawater, and they break open. And so all of these plates that are forming these coccolithophore algae are released and reflect the light. And in fact, this is what you see in the famous White Cliffs of Dover in the southeastern part of England that are all there because of virus infections. So you can see virus infections literally from space. So not surprisingly, the fact that you have so many of these viruses and they're so prevalent in the ocean ecosystems, they're also very important for just fundamental biogeochemical cycles which happen. So we all know about fossil fuels and deforestation. That's a big issue, of course, as well. But there's a natural carbon cycle that happens as well. CO2 is released into the atmosphere, it's taken up in the surface waters, and will just generally circulate. Well, for a long time, the oceanographers were actually very confused because what they saw was there was a lot more carbon present in the upper ocean than they expected, because they knew how cells were growing, and they expected that there would be a certain amount of dissolved organic carbon, this DOC or particular organic carbon, but there was far too much of this dissolved organic carbon than they expected. And the only way to explain that is the presence of viruses, the viruses killing off the cells like they did with the coccolithophores and making all of those calcite plates to then drive a lot of the carbon cycles in the upper ocean. So the presence of viruses on our planet really is driving a lot of the biogeochemistry. And of course, well, maybe this could be what's happening on other planets as well. It's not just the oceans and algae which are infected. Basically, viruses infect every organism that we have found cellular on Earth to date. This is the big tree, the ribosomal RNA. This is what's conserved in all cellular life. So you can compare each of these to each other. I mentioned the eukaryotes, the archaea, and the bacteria before, the 3 main groups of life. Just for reference, we are here. That actually includes all the animals in this one little twig here. Sulfolobus is here in the archaea, bacteria, including E. coli as well. Every cellular organism that we found has a virus associated with it. And they're present in the most extreme environments, as you heard in the introduction a little bit earlier on. So this is one of our favorite environments, Chino Ike Jigoku. Chino Ike Jigoku roughly translates from the Japanese as blood pond hell. And the reason for that is it's very high temperature. It's about 75 degrees Celsius, this particular lake, and the pH is 2. And it's got lots of cells that live in there, and not surprisingly, viruses which live on those cells in these environments. This is the one, again, which was mentioned in the introduction, a virus that I've been working on for over 20 years now. This is the sulfolobus spindle-shaped virus. This really beautiful shape, which actually doesn't look like the shape of what most viruses are, may have something to do with this particular extreme environment. And this has all been a collaboration with a wonderful researcher at the University of Texas Medical Branch, Mark Murray. We published this a few years ago, and as all of us know now, you usually look at a PDF, you don't look at the cover of the journal anymore. So if there's you get your picture on the cover, you've got to use it in any one of your talks. So absolutely everywhere, very interesting shapes. But what about, and important for the environment in general, but I'm going to try and convince you in the next couple of minutes that viruses are actually good for us as well, just personally, above and beyond what's going on in the environment and various cycles which are happening there. The first of those is if you look at the human genome. In the human genome, there are about 8% of the genes which are very clearly derived from viruses, these retroviral-like elements. So 8% of all of our genome have these retroviruses that have just inserted themselves into the genome in our ancestors. There was a viral infection, got into these cells, it's popped itself into the genome. And no one argues whether 8% of the genome, our genome, was really viral. And this is not just true for humans, it's true for really many other organisms as well. About another 30% of the genome are a bunch of repeated sequences that are present in the genome, which might have come from viruses. Whether they have or not, that's sort of an open question. And just for the comparison purposes, the protein coding genes, or what makes humans humans, is only about 1.5% of our genome. So we're more viral than we are human from just looking at our genome. Now, maybe it's junk DNA. I'm sure all of us have heard about junk DNA. Well, it turns out that not all of these virus insertions are junk. One specific set of genes that was inserted into the human genome is absolutely critical for motherhood as we know it. There's a gene called syncytin, which is in all of our genomes, and actually in the genomes of all placental mammals, which is absolutely required for the formation of the placenta. In the placenta, you have the developing fetus, and the mom have to communicate with each other through this really amazing organ. And some of the genes that are involved in that are actually derived from viruses. So there was an infection of the ancestor of all mammals hundreds of millions of years ago, which then was maintained, allowed the development of placenta. Otherwise, we'd all be coming out of eggs from that. So this, viruses basically created motherhood. So we wouldn't have motherhood if we hadn't had virus infections. Even more bizarre, at least to me, is it turns out that viruses are also important for memory formation. So again, these are viruses which inserted into our ancestors many millions of years ago and are actually used, the viral structural proteins that look a lot like those virions I was showing you right at the beginning, package little pieces, probably mostly RNAs, which then move from nerve cell to nerve cell and are important for memory development. So virus is important for motherhood. Virus is important for memory, although my memory seems to be going, maybe I need to get infected by a few more viruses. So these are just general things that we have, but also the understanding of viruses has allowed us to make huge amounts of progress in terms of using viruses to cure disease. So these are not viruses causing disease. These are viruses curing diseases. Almost all gene therapy, where you take, have a defective gene of some kind, you're trying to repair it. Almost all of these are using various different viruses, adenoviruses, retroviruses, et cetera, And one of my favorite examples of good viruses is using a virus that infects a bacteria that would otherwise be making you sick. So you use the virus that infects the bacteria to kill the bacteria, and in that way, you're not getting sick from the bacteria anymore. And this is something called phage therapy, which I think is actually going to turn out to be particularly useful, particularly in this day and age where we have huge amounts of antibiotic resistance in bacteria. But the bacteriophage have been evolving with these bacteria for millions of years. They're really good at killing off the bacteria. So disease-wise, potentially very, very useful. And then another example of really, I think, amazing virus, excuse me, being good is there's a particular plant. It's called panic grass. I'm not sure, you probably have it in Mexico, but you definitely find it around a lot of the hot springs where we do our sampling in the US. It's called Dicanthelium lanuginosum. If this plant is infected with a fungus, and if that fungus is infected with a virus, it can grow at 55 degrees Celsius. If the plant doesn't have the fungus, it doesn't have the virus, then it can't grow at these high temperatures. So it's that virus infection which allows the plant to grow under conditions where it otherwise would never be able to. So yeah, again, this is another example, I think, of a really good virus. Another good virus, although depends a little bit on their point of view here, are viruses which infect a lot of the now not eukaryotic algae that I showed you the picture of infecting off the coast of Cornwall, but a lot of the other microbes in the oceans, the cyanobacteria, which produce up to 20% of the oxygen that we breathe on any given day. These then form oxygen through photosynthesis, just like plants are making oxygen in our atmosphere. But the process on how they make oxygen is dependent on photosynthesis and all the proteins that are involved in photosynthesis. It turns out that there are viruses that infect these cyanobacteria, the bacteria that are producing oxygen, which carry with them genes that are important for photosynthesis and allow these cells to grow in conditions where they otherwise wouldn't be able to. So virus infection of some of these cyanobacteria are really important just for the amount of oxygen that we have on our planet, and maybe what kind of oxygen you might have on some kind of extraterrestrial planet as well. And there's again, still a little bit controversial, I think, but the great oxidation event, when the amount of oxygen really took off on Earth, maybe that was dependent on some of these viruses. And so this brings up the immediate question of, well, how old are viruses in the first place? Where did they come from originally? So you remember, our definition of viruses is they've got to, get inside a cell and use cellular machinery in order to be able to replicate. Well, okay, but what about before cells? Were there viruses before cells? Well, we couldn't call them viruses anymore because they wouldn't be infecting them. So number of people have done some really nice studies, particularly Mart Krupovic, Valerian Dolja, and Eugene Koonin, published a nice review in 2019, and I think there's a new one coming out very soon, where they think about the origins of viruses. If you think about origins of life scenarios, almost always you end up with parasitic replicators. And this is all simulation, but certainly it looks as if some kind of self-replicating system that will be like a virus, but is not dependent on cellular milieu. That's probably how virus-like genomes started. And then they probably infected some of the very earliest cells and picked up these virions from the outside, and then just over time have continued to co-evolve. Now, this is all really theoretical, but I was very fortunate to be able to contribute to some of this in studying some of the viruses from hot springs. This is from Yellowstone National Park. This is me about 20 years ago, slightly less gray hair, collecting some samples from this hot spring, 85 degrees Celsius and pH of 3. We're actually looking for those lemon-shaped viruses But instead, we found something really different, which we now call sulfolobus turritid icosahedral virus. Beautiful structure. I was probably the first person in the world to see this, which is really amazing. And then Liang Tang and Jack Johnson at the Scripps Research Institute solved the structure of this particular virion. And they said, these really neat sort of knobs coming out of the outside of the virus are really cool. But what's actually even more interesting are the individual proteins that are put together to make up the rest of the virus. And what they saw was really fascinating. And that is that the structure of this virus from the hot spring that infects archaebacteria has a structure which is basically identical to a structure, now a molecular structure, of a virus that infects bacteria and a virus that infects eukaryotes, the three completely different groups of cellular life. So the structure is identical, but the sequence is completely different. And so how could we explain this? One possibility is what we call horizontal gene transfer, but that means that a virus would have hadn't gotten from a bacteria to an archaea to a eukaryote. That seems really unlikely. Potentially convergent evolution, because these structures, the icosahedral or more or less spherical structures, pretty well conserved in viruses. But we like to think this is an example of a virus that infected that common ancestors. I showed you one of those previous slides that Kunin and Krupovic were talking about. So this structure has been maintained over evolutionary time, probably billions of years, that bacteria, archaea, and eukarya separated from each other. And so we think this is an example of a structure which has been conserved for a really long period of time. And again, probably then indicates that there were some virus particles that had these similar kinds of structures back in the very origins of life. We published this in 2004, and in fact, we were said to be like Darwin, which I think is about as good as you can get as far as a biologist is concerned, to be compared to Darwin in the literature, which is really fun. And then the structure was also solved soon thereafter. But then the question came up is, okay, all of this stuff is indirect. Okay, we got these structures today. We've got these theories on how things could have worked. Well, how do we know that there's been life on Earth for billions of years? Well, we've got microbial fossils. Okay, that's great, but what about virus fossils? Are there any fossils of viruses, or more to the point, any fossils of virions? I was like, that's a really interesting question. And are there fossils of viruses on Mars? Could we find them? The question, of course, is, first, are they there? Do they even exist? Or do they exist and we just don't know how to detect them? And so this is where Dr. Jim Laidler, wonderful, I would say typical graduate student at Portland State University, shows up in my virology class one day, knows the answers to more questions than I do, comes in my office one day and says, I'm actually a medical doctor, but I'm kind of having a midlife crisis. I want to get a PhD. And so I said, sure, yeah, come work with us. So I convinced him to work on this virus fossil project and basically say, okay, are there virus fossils? And if they're there, how can we detect them? And this is not a completely random idea, because if you think about volcanic hot springs, so where we're working on, where we find these lemon-shaped viruses, where we found that virus with the structure that's really well conserved in different organisms, what volcanic hot springs you have is you have superheated water at depth, heated up by the mantle of the Earth. It comes up through the rock, it leaches out all the minerals that are in the rock, then it gets to the surface, it cools down, and all of those minerals precipitate on whatever is there in that particular environment. And I'm sure many of you have been to hot springs. You see these wonderful terraces, all of the accumulated minerals which are happening there. And so we said, okay, let's look at some of these volcanic hot springs and see if we have any kind of mineralization of viruses. The geologists tell me they're not fossilized, they're just mineralized. And so we did this, mostly Jim, and looked at a number of different environments where we could see this rapid mineralization taking place. And we also said, okay, we'll look at these environments, but what we really want to know is what's actually happening in a controlled situation. So we did some of these experiments in the lab, and we specifically looked at this virus, bacteriophage T4, because it has this really wonderful structure. And what we saw in that wonderful structure was we could get a really nice silica coating around the outside. Silica is what we started with. And the same thing is true for some of these spindle-shaped viruses and also for some of these rod-shaped viruses. And a little bit easier to see, again, I've got to show off that we're on the cover of Astrobiology. We actually just have a transmission electron micrograph with a silica-treated bacteriophage and a untreated bacteriophage, and you can really clearly see the difference here. Unfortunately, as far as virus fossils are concerned, this really nice silica coating, after a couple of days, turned into a real amorphous blob that we couldn't detect anymore with our techniques. So that was kind of a disappointment as far as the student was concerned in terms of finishing their PhD. But this actually turned out, and I can talk a little bit more about this later if people are interested, was how we ended up founding the company. And the reason for that was that this mineralization process, we found kind of by accident, is reversible. And the mineralization actually protects the virus and also protects vaccines so that they can be stabilized. And so that's the whole process, the company, et cetera. That's a whole nother talk in and of itself. So that's mostly what I wanted to talk about for now, but I did want to quickly, and I think I'm actually going to have to stop my share here because I think I shared it incorrectly. So I'm going to share my screen again, and we'll take a quick look at here we are. One more, just a brief video that was put together a couple of years ago, and most of it's really good. Just ignore the part that comes at the very end. So let's move to there. Next slide. Brief video here.
The reason that NASA should be really interested in viruses is that, at least on this planet, there are more viruses, 10 to 100 times more viruses than there are of anything else which is around. The question really is what about humans in terms of, are humans viruses? lots of viruses that we know of actually change their environment, change their host in particular. And my favorite example is actually those viruses that infect some of the algae and the bacteria in the oceans that make the oxygen we breathe. But you think about it from the human Earth perspective. Humans have clearly changed what's going on Earth, and changed it in such a way that it may actually be beneficial for the Earth, and certainly beneficial for humans. And so you can think about all kinds of things that have happened on our planet which may actually be positive from an Earth point of view.
Hi, I'm Dr. Ken Stedman from Portland State University, the Center for Life in Extreme Environments.
So not actually coming soon in 2012, maybe 2021, if you switch around those two numbers. I'm realizing that filmmaking actually takes a very long time. So before we finish up, I did promise I would tell you something about viruses in space. And lo and behold, here are viruses in space. Astronauts, us, we've got viruses in our genome. We're infected by viruses. So there are already viruses in space. We know that they're there. But what about further than low Earth orbit, et cetera? And what about just thinking about viruses in space from an astrovirology kind of point of view? First is, as I mentioned at the beginning, these have very unique structures. The virions, they've got these kinds of structures. They've got lemon-shaped structures. Is there a way we could actually develop some instrumentation so that we could go to Europa or Mars and actually detect these virions that are there? And everything we know about viruses now says that viruses have to infect a cell in order to make more viruses. So if you found viruses, then that would be a pretty good indication for life, I think. And one of the things I like to do is sort of flip it on its head as well. If there was an alien intelligence that was coming to Earth and was looking for life, what would it sample? It would sample our oceans, because clearly most of the planet is oceans. What's it going to find in the oceans? It's going to find mostly viruses, because that's mostly what's there. All those little dots that I showed you right at the beginning. So, it would think we're a planet of viruses. I would agree. We're a planet of viruses. In fact, Carl Zimmer wrote a really nice book called A Planet of Viruses, which gets into this a little bit more detail. Other questions that are very interesting from an astrobiology perspective is, how do viruses fit into the origins of life and life on Earth as we know it? Some people have theories that viruses might have been involved in the invention of DNA. So we went from an RNA world to a DNA world. Maybe there were some viruses involved in that process. We clearly don't understand very much about the good viruses. We all, unfortunately, know far too much about the bad viruses, but there are certainly some good viruses out there. What happens in the space environment when you put viruses there? This is really important for exploration. Going to Mars, what happens to viruses in the space environment? Those could be the plant viruses that are important for whatever kind of nutrition that we're putting onto some of these, deep space flights. What about the astronauts, et cetera? Viral ecology, what's going on, just not on our planet, but again, on other planets, et cetera. We're probably bringing viruses with us to Mars. Are viruses coming back? I think that's going to be a big issue, but if you're bringing back samples, they're supposed to be bringing back samples from Mars. What should we be doing about those samples? Could they have viruses on them? I don't think it's a big issue, but it's certainly something we should be considering. So that's all I really have time to talk about today. If you're interested in more about astrovirology, myself, Kathy Bywaters, Gary Trubel, and Penny Boston, who I think has been involved with the Mexican Society for Astrobiology in the past, we put together a meeting without walls, and all of that is recorded at the NAI. So if you're interested in more details, there's a couple of days. We did a couple of half-day meetings on that. And so hopefully I've been able to convince you over the last 45 minutes or so. Viruses have a bad rap. They're not all bad, really. And they're absolutely everywhere. Some viruses are clearly good. Older viruses, I don't think we have a really good handle on that, but they're probably really, really old. And we have some viruses in space now, again, because we've got astronauts. But what about other viruses in space? Are there more there? And a lot of this was discussed in another cover that we got in Astrobiology, a review article in 2018. And that's what generated that killer alien viruses thing that I used at the very beginning. Clearly, they did not read our article when they looked at that. So with that, just wanted to advertise a little bit some of our sampling grounds. In the movie, you actually saw me sampling from this place in the background here. It's a place called Boiling Springs Lake in Northern California. This was our group last year in the time of COVID. Usually we have about 15 or 20 people. There were just 4 of us this last year. You see, we're all doing our good protective equipment. Our work has been sponsored by NASA, particularly actually the virus fossilization work, National Science Foundation, NIH, University of Texas Medical Branch, Gordon and Betty Moore Foundation, Portland State University. And again, if you're interested in getting more answers to some of these things, check out our website, gets into more detail. Feel free to send me an email or follow me on Twitter, where I mostly talk about vaccines these days. So with that, thank you so much for your attention. Thank you for the invitation. This was a lot of fun, and I'm happy to try and answer questions if I can.
Yeah, well, Dr. Stedman, thank you so much for such a great talk. So then hopefully we have a lot of questions we are streaming in Facebook and YouTube and Zoom. So let's try to figure it out first. Let's go with the Zoom, as I understand. So if anybody has any question, so please just unmute yourself, aprieten el botón del audio para que se escuchen y hagan la pregunta en inglés o en español, como quieran, y aquí les ayudamos con el inglés.
Bueno, yo levanté la manita porque Luis Miguel no tiene su audio, tiene su audio abierto, entonces no sé si quiere hacer pregunta también, pero voy yo. Okay. I'm going to do it in English, sorry.
Easier for me.
I suppose you are familiar with the minimal life concept. So I wonder if you would think that viruses are something between inanimated stuff and life, and how do you support, if you think that may be possible, how do you support that?
Yeah, so that's a wonderful question. And I think the answer is, and whenever we say it's a wonderful question, it means we don't know what the answer is. But I think, again, given what I've seen and a lot of the modeling, is that as soon as you start to have some kind of cellular life, you're probably going to have some kind of parasitic replicators that are associated with it. And even this has been seen in some in vitro evolution experiments, even prebiotic kinds of chemistry. You see some of these cheaters. Now, do you call those viruses? Do you not call them viruses? It really kind of depends on what your definition of a virus is. And so now our definition is that they have to be infecting a cell. But is a parasitic replicator a virus? Now, one of the things is, we humans like to name things. The things that do it, they don't care. They just do it. So I think that the short answer to that is, I think that probably very early on in the evolution of life, it's highly likely there were at least virus-like elements that were present there. Whether we'd recognize them now as viruses, I'm not sure. But I think that thinking about some of these processes are going to be very important in thinking about some of these very early life models.
Okay, well, while someone else is just motivated to questions, so I have here a message from Ralph
Looks
Miller.
like Luis has his hand up.
Let's see. Okay, Luis Miguel Rodriguez. Exacto. Go ahead.
Okay. Good afternoon, Mr. Stedman. Thank you for this conference. It's very interesting, really. And I have this situation. This is a hypothetical situation. In the past, Mars had the condition to support life. If so, it is possible that viruses exist on this planet. Mars is currently a place that cannot support life, but viruses may still be there. Faced with this possibility, should astronauts take precautions against the possible presence of these Martian viruses?
So, wonderful question. Thank you, Luis Miguel. All viruses, and I didn't mention this very much, but viruses generally have very narrow, what we call host ranges. And so they only infect a few numbers of organisms, and those organisms are really quite closely related to each other. And okay, we'll think about SARS-CoV-2 now, right? It infects lots of different animals, mammals that are all actually on a molecular level very closely related to each other. And so astronauts going to Mars, I think if there are any viruses there that would be able to infect them, that would assume that there were human-like hosts on Mars that length of time ago. So I'm actually not too concerned about that. What I think is actually more likely is that there was some kind of microbial life, and there may have been then some microbial viruses that got associated with potentially life that was present on Mars. And those kinds of things, I think, are things we might want to actually be, if anything, more concerned about, because, how similar are the Martian microbes to whatever human microbes we might have.
Okay, thank you.
Yeah, you're more than welcome. Thank you. Great question.
Muy bien, ahora Miguel, vas.
Thank you very much, Ken, for accepting the invitation. It was great. I will actually need a few weeks to digest the material you just showed. But I am curious about something related to this contamination issue. We have this spacecraft that have landed in Mars, but there are also other spacecraft. Actually, we did have a talk back in December from Jason Burns in Idaho about Dragonfly that will land in Titan. So here the curiosity I have is, we know these spacecrafts, when they are assembled, all the subsystems have been created within very clean environments. Is there any test, thinking about contamination the other way around, do they go through tests for virus, all these spacecrafts, or would you think that is required? I mean, just to avoid contaminating some other places in the universe.
Yeah, wonderful question, Miguel. And I sort of briefly sort of mentioned that when I said planetary protection at the end of my talk. I think it is a very important thing to think about is, wherever we go, we're bringing our viruses with us. And in those clean rooms, they do find some bacteria, not a huge amount, but still some. In fact, finding viruses is harder than finding bacteria. Partly because they're smaller, that's not surprising, but also you don't have the same kinds of ways of discovering them, like the 16S rRNA. Again, these sequences which are conserved in all cellular life, you can use the polymerase chain reaction, amplify them, get lots and lots of that. there's nothing in viruses that's the same. And so in order to see what viruses you have, you really have to sequence all of the nucleic acid that you have there, all the RNA, all the DNA, and there's not very much of it. So it's actually really hard to test some of those things. But I know that they're doing it. I know they're actually trying to do some of these things, but it's only really very recent technology that's allowed us to do this. And these kinds of things, were not done for some of the rovers even, just because we didn't have that kind of technology. We knew that it had very small amounts of bacteria that were associated with them, but we don't really know that much about the viruses. So it's a very, I think, important and interesting thing to think about. But getting back to Luis Miguel's question, I don't think that it's likely there's too much life there that those viruses could infect. But I think it is important to just see what we have. And I know that people at the Jet Propulsion Lab, where they have these spacecraft assembly facilities, are definitely looking at these things.
Thank you.
Okay, so then just making a few minutes more just for anyone else sending any question. I'm going to read now a few comments from the YouTube channel. So then this one is from Ralph Mueller, I guess. Hey, Ken, great presentation. Very regards from Germany, Ralph.
So if I can comment really quickly on
sure,
Ralph. So we're actually trying to set up a collaboration to do some of the experiments that we were talking about. So
okay, great. So really happy that also, I mean, I am in Germany too right now. So then it's great that there are more people besides Mexico. Okay, so then also Lucia Gonzalez, thank you so much for sharing your investigation, doctor, and congratulations. Okay, so then here we have one question. This one is from Jaime Leonardo Moreno. Hi, great talk. Do you think that there are more viral shapes to find? And the second question will be, do you happen to know when was the last time we run into a phage with an unknown shape?
Okay, so the first question is, do I think we can still discover more structures of virions? I think the answer is yes. We have studied so few environments. Now, most of them are going to look like these bacteriophage. So yeah, that I think the vast majority. But there are going to be a few which are going to be very different. And again, something like the ones that we found and some of our colleagues have found as well. But people just haven't looked, I think, in many cases. And so we don't know. And I think these extreme environments are going to be a really good place to find them. Now, why they have these very different shapes in extreme environments, I don't think anybody knows. It's a really interesting question. And I guess the second part of the question, if I understood it correctly, was, how recently was the last one with a really new structure found? The answer is I don't know. But some of the group, particularly the group of Mart Krupovic and David Prangashvili at the Institut Pasteur in Paris, they've discovered lots of new viruses with different shapes. Again, almost all from these extreme environments. If I remember correctly, a couple of years ago, they published on some new kinds of structures. One of the things that we're actually very interested in is looking at the structures of these, the SSVs, the lemon-shaped viruses, and trying to understand how they fit together. Because if you think about virus structures, of virion structures, I should say, is they're all repetitive in some way. And they're either sort of a helical spiral or they're icosahedral, a little bit like a soccer ball and also like a spaceship, because if you think about the geometry, how do you have the largest volume with the smallest surface area? You have a sphere. And the easiest way to get a sphere with repeating units is an icosahedral, a lot like a soccer ball. And so it's understandable you would have helical structures and spherical structures, or icosahedrally symmetric structures. But what about these other structures? How will you get these different structures? And it turns out that for our virus, again, this SSV virus, and I think I tried to bring a I've got some printed models of them somewhere, but the 3D printer, I'm not finding the one I'm looking for. But what we think is happening, so what we thought when we published that paper in Virology in 2014, we thought that the structure, this sort of lemon shape, was actually more like the interior of some of the shapes that you see in things like HIV. So the internal structure of HIV is a little bit like an icosahedron, something called a fullerene cone. So that's what we published. We're probably wrong. We have some new data which actually looks as if they're made as spirals. And so the structure is actually a whole spiraling structure. So we're very interested in trying to figure out, what this structure is at a higher resolution, but then also how the pieces fit together, because this may tell us something really fundamental about how proteins fit together and how you get stability just at a structural level.
Okay, so then we have two more questions. One is from Nabor Lozada. So, hey, fantastic talk. The question, we know that all cellular life has been infected by viruses. So if we find life in another planet, so can you imagine that this kind of alien life could be existing without the presence of viruses?
So great question. I can't imagine life on this planet without viruses. So it's kind of turning the question around. And we actually had a thought experiment, a whole group of us a few years ago at a colloquium, the American Academy of Microbiology. Basically, we said, what would life look like on Earth if there were no viruses? And we said probably a kilometer layer deep of slime. That's about all of the life we would have on Earth without viruses. Now, the flip side, again, is alien life, would it have viruses? I think a lot of stuff can depend on what that alien life looks like. I would actually be really interested if it were life that didn't have viruses. I think that would be really fascinating as sort of a different way that life is evolved relative to life here, because life here clearly has evolved with viruses. So I think that either answer would actually be really interesting. I would expect personally that there would be viruses associated with that, again, given the predictions in terms of origins of life. But if they didn't, actually be really fascinating, it'd be really interesting to see what differences, how do they manage to do some of the things that we've used viruses for.
Okay, the next question is from an unknown name, but that is like 1456. So
okay,
it's a very good question. So will it be possible to sample the space for virus on Earth? We can pump air into a filter to trap viruses. Is there any effort to do something like this in a space where there is no air?
So basically to filter whatever you have in space and then maybe concentrate and get something which is there. That's a really interesting question. Now, there's very little matter in space to find in general. So I think you'd have to concentrate very, very large amounts. Even on Earth, we've studied the virus you mentioned in the introduction, the one which is recombination between DNA and RNA viruses. Interested in more on that, talk to my associate scientist who does speak Spanish. I'm sure he can give you a great talk on that. But there was a study that was done in Korea, really great study, where they were looking at viruses they found in different environments. They took air and they took 50,000 liters of air and concentrated it down and then looked at the viruses they found there. And it turns out they actually had some viruses similar to the ones that we're working on. So I think, given that idea, again, of going out in space and just collecting really enormous amounts of volume, which is really what it would be, and then trying to find something in there. I think the needle in the haystack analogy doesn't even come close to trying to find some of these things. And one of the things that we actually do when we're going and looking for viruses in the environment, we don't actually really find many viruses directly. We only find them if they're infecting something that we're working with. So those lemon-shaped viruses, the spindle-shaped viruses, we only find them because they infect sulfolobus. There are undoubtedly other viruses there in these extreme environments. And I guess back to one of the previous questions, are we going to find new viruses with new structures? We're specifically looking for the ones that are infecting cells that we can work with. And that means that we're not actually looking at all the viruses that are there. So I think, going out, filtering massive amounts of space or even the upper atmosphere, what would you find? And people have done some things in the upper atmosphere, so getting way up above the Earth, and you do find some virus-like particles very high up in the atmosphere.
Okay, so then I have two questions, but just probably making a few minutes more before closing this session. So then the first one is a little bit basic, silly maybe, but I'm really wondering what actually are doing viruses when they are not infecting cells, right? So then it's just like you have this awesome work showing that, well, they just silificate, and then this mineralization, and then they stare around. My question is a little bit technical, but then when you say that there is this reverse activation, inactivation based on this mechanism, are we talking about a specific, like total remove from the silicates, or is just a rate? And for how long is a virus actually can stay in that state, like kind of latent state or normal state?
Okay, so that's a great question. And so what we showed a couple of years ago now is that when we do the silicification, the way that we do it, pretending basically that it's a hot spring, we get infectivity that we can get back up to a couple of months later. So months, not years, not decades, not centuries, not millennia, etc. So it's a relatively short period of time. For vaccines, that's great. For getting a virus from Mars to the Earth or Earth to Mars on a meteorite, that's a little tougher. So at least the way that we're doing it, I think that stability, still being infectious, is only going to be on the order of a couple of months to maybe a couple of years if we tweak the system properly. But I think much more than that, it's unlikely. That being said, now that was our system. There are people who have looked at viruses that you find in sediments, so underneath lakes, and particularly in this case, it was the Black Sea, where you can date how old these sediments are. And they found some viruses in some 1,000-year-old sediments that could infect cyanobacteria. So, that's getting to thousands of years. Now, admittedly, they knew they were there. Again, it was an oceanic environment, cyanobacterial phage, those kinds that you would expect. But yeah, how long they can actually really last, and there may be other conditions. I'm actually talking to some of my colleagues about maybe some conditions where we could get viruses that could last for longer. We'll see.
Okay, and then going to my last question, trying to see if someone else has any other question. So it will be like, I am going to put you in a little bit of troubles, Ken. So then, because at the end, viruses are just stealing everything from life, right? Well, from cellular life. So then when you are talking, because you are in this amazing research group at NASA, so then trying to identify and validate biosignatures for viruses. So have you find or have any idea of which kind of features in the viruses, proteins, genetic material, what particular could be a good sign, kind of candidates for biosignatures?
You're right. You're putting me on the spot. The answer is I don't have a really good answer to that. But one of the things to think about, and again, I'll go back to my nice little 3D printed version of our bacteriophage here. The morphology here, it's really hard for me to imagine how you could get this abiotically. So if there's some way to get a morphology that was, this kind of shape and size, I think that would be a really good biomarker. Now, that's tough because we don't necessarily have the resolution on some of the techniques to be able to do that. And we're actually talking to people about Secondary ion mass spectrometry, can you get to some of the levels? And maybe you can actually get elemental analysis. And if you have a rock that you're looking at, can you scan, do you see an icosahedral structure in it, which is made up of the appropriate elements, or better yet, the appropriate chemistry? And that would be a potential biosignature. There's also another way of thinking about biosignatures, which is kind of a complete opposite one. And that is one of the things that I didn't mention, is I talked a little bit about the viral proteins that change photosynthesis. So, and there are other proteins that are brought in on viruses and then change the metabolism of the cell. It's possible that these enzymes actually work differently enough from the cellular enzymes that you could identify an enzyme maybe by a isotope ratio that was then a viral isotope ratio as opposed to a cellular isotope ratio. So that's a possible way of thinking about it. But a biosignature of a virus is tough. I already talked about the biosignatures in the cliffs of Dover, because you have all this viral lysis. But yeah, those are really, really challenging things to do, and probably people smarter than me can come up with some good ideas on how to look for those.
Okay, Antigona, please go ahead.
Yeah, I was thinking on what you said about the definitions, because, okay, so we have the definition of virus, that is this structure that contaminates life well, it gets stuck to life, yeah. Just to reproduce itself. But then basically the way to search viruses is it's in the cells and not as a free something. I'm not going to say free life. Yeah. So it could be possible that these structures could be free without infecting, without having to get into a cell. I am trying to avoid infect.
Yeah, because no. Infect is fine.
So is it possible that we are just not looking for those structures that are able to reproduce themselves without having a cell?
Okay. So the question, sorry, I think I'm following your question now. So the question, do you have something which is self-replicating in the absence of a cell? Something which is then going to self-replicate.
And we don't find it just because we don't look for it.
We're not even looking for it. Yeah. So all viruses that we know of now, again, this is just from our own point of view, they all depend on some kind of cellular machinery. So fundamentally, viruses all have nucleic acids and they have proteins. Some of them have lipids as well on the outside. And so those are the same molecules that the cells are using. And making those molecules from the individual elements and then other chemicals, these are things that cells are doing. And viruses, to a different extent, some viruses do more, some viruses do less. All of them are taking advantage of some of those things that cells are doing. So maybe, again, this is hypothetical, that a cell throws something out, it's garbage, or whatever else cells do. And then something else in the environment could pick that up and maybe use it. Maybe. I think you end up with it, again, getting back to your definition thing, is what's a cell, what's a virus? And how you differentiate between these things is really hard. If you think about an intracellular bacterium, okay, well, there are chlamydia, there are other things that can only survive when they're inside another cell, but we call them cells rather than calling them a virus. So, and we have really tiny cells, spores, a spore, which is, just hanging out, doing nothing. It can only then make more of the bacteria if it gets into the appropriate environment, and then can make more. So, what's a spore? What's a virus? What's a cell? I think lots of these things, and they start to get, actually, the definitions start to get really tough, because I mentioned right at the beginning, very small. This is how viruses were originally discovered, but there are what people are calling giant viruses, or it's the official term is large nuclear cytoplasmic DNA viruses, but giant viruses that are bigger than some cells. There are viruses that are bigger than E. coli. And they bring lots of genes with them, but they can't replicate unless they get inside another cell. but that's the environment that they're in. And so depending on the environment, so it's kind of like in my video, it's like, are humans viruses, taking advantage of a different, it depends on your definition, really.
Okay, so then just a very last one, Ken. So then I would like to take the opportunity of the advantage of your experience as a very great science communicator in public social media. So, and also because very probably if I am NASA and I find, any sample or whatever, any weird thing there, so you are going to be one of those guys they are going to call, and so then I also saw your videos, your interviews in the beginning in different phases of the current COVID-19 pandemic. So, and I also saw how we're changing your opinion regarding how much dangerous or serious was the pandemic and all the rules and everything. So then, as a scientist, how do you think will be important to communicate to humanity? First, from the social point of view, if we find alien life. And second, as a virologist, when is the point when you are going to give up and say, yes, this is cellular or something kind of life or a viral kind of life or biological system?
So you always ask the hardest questions.
Sorry.
Yeah. So what's a virus and what's a cell? I think those are things that are very, very difficult to answer. I'm going to answer the second one because it's a slightly easier question to answer. And at least right now, all viruses depend on translation from their hosts. And so if it's got a ribosome, so something that takes RNA and translates it into protein, all viruses are dependent on the cellular translation machinery, all the viruses that we know of so far. Now, if there's a virus which we find that has a ribosome in it, okay, what do we call that then? So I think that's where I think some of this is really, at least for me, that's where these things are going to get really blurry. There's also some arguments about energy transduction, energized membranes, etc. But for me, at least right now, ribosome, and that seems to be the thing which is really different. And potentially the ribosome and translation was also really important in terms of life's origins at the very beginning anyway. So I think that makes sense from that point of view. How to think about, communicating, we have life, we don't have life from a sociological point of view. I'm actually going to defer on that and ask the sociologists and sort of say, this is, our science, science says, this is what we see, this is the evidence we have. And, certainly the best explanation that we have, given the evidence that we have, is, X, it's alive or it's not alive. But you have to be careful again. It's how you're interpreting the data. That's what all of us do as scientists. We interpret the data that are available to us. And then, going beyond that is, okay, with this data, this is my interpretation of it. It's not really answering your question, I know, but.
No, it's perfect. It's perfect. It's good. I mean, it's important for people also to understand how difficult it is for scientists to deal with this, with the data, with the hypothesis, and finally with what we want to communicate to public.
Yeah, no,
so
it's not easy, and practicing a lot and making a lot of mistakes. I'm really good at making mistakes.
Okay, so then I think we can close now. If I don't know, Miguel or Antigona tells me if I am missing any other question or something like that. Okay.
Well,
so then
just I mean, if we are going to close, just a comment.
Sure.
Actually, what I would like to is a thank you comment for the people who follow us through YouTube. Thank you very much for being there. And also thank you to the SOMA members who were with us in this great talk by Ken. And of course, thank you very much again, Ken, for accepting the invitation. We will keep in touch as soon as I learn a bit more about viruses.
Oh, so thank you so much for the invitation. This was a lot of fun, and great questions, really good questions. Greatly appreciate it, and looking forward to hearing more of them.
Hopefully when we are back to some kind of normality, so then we manage somehow the society just to invite you physically to Mexico, and then you can enjoy, some food, and also talk to the students in Mexico. They are super smart, more than us. And then I'm sure that you will get fun too, and make us a lot of collaborations. Antigona?
Thank you very much for coming.
Yes, thank you. Thank you so much. This was a lot of fun. And have a good weekend.
Yeah, so for everybody, and also for the people that is still there, just remember that it's coming que viene el tercer Congreso Latinoamericano de Astrobiología, que se va a llevar a cabo en México en agosto, del 3 al 6. Es una conferencia virtual. Sí, todavía existe fecha para inscribirse. Las cuotas de inscripción para los estudiantes son muy bajas, son justamente para que se animen a asistir, y pueden encontrar más información en la página de la sociedad www.soma.org.mx. Y con esto